viroids, prions and viroids. We'll just very shortly discuss about the viroids at the end of this particular presentation. These viroids are some plant pathogens. Till now, no viroids were identified to cause any disease in animals and human. These are, let's say, again, the primitive forms. Even their conformation is simpler than the viruses. We know that the virus is the most simplest form, but even the viroids, they are much more simpler than the viruses, because they don't have any proteins. They are only the nucleic acid. So they are the RNA and nucleic acids, and they can produce some disease, and they can be transmitted from one to the other. So that's more important for the agriculture people, because the viroids are associated with some infections in plants. Very common infections. One picture I will show you, probably everyone will recognize. Yes, I have also seen that particular disease. So that we'll not much discuss about the viroids, but we'll be focusing on the prions. Of course, don't get it confused. We cannot mix it, the prions and virus together. Prions are totally different. So as you know, the prions, Stanley Prusiner, he got the Nobel Prize for his discovery about this particular compound, I should say compound. We can't say it's an organisms, the prions. So, but this is associated with disease conditions, and that disease are interestingly seems to be transmitted, but look like exactly an infectious kind of a thing. But this is not into that category of discussion, because this is a purely different type of phenomenon that occur. These are certain proteins, simple proteins, no traces of any nucleic acids, simple proteins, and that particular defective protein convert the normal protein into a defective one. So when the defective protein go on accumulating in the body tissues, that lead to some pathological consequences, and that too also and non-inflammatory pathological changes. It's quite interesting. Whenever we say it's a pathological changes, we say that the infectious organism causes some inflammatory reactions, but here there is no inflammations occur. So of course the disease has been detected in animals from quite earlier times, particularly the scrapie. Scrapie is a particular kind of a disease that occur in sheep, which has been reported long time back. Even in 17th century, there is description of the scrapie disease in sheep, but nobody could able to explain the causative phenomenon of scrapie, which is an neurodegenerative changes leading to mortality or fatality, terminal fatality. So the disease descriptions were there in all literature for quite a long period, but later on it was this Stanley Prusiner, he put forward it as what basically happened in this kind of infections, and he designated this infectious protein particles, proteinaceous infective particle, and he given the name as prions. Okay, and this prions, for easy understanding, universally people use this particular way of writing the prions as PRP-SC as a superscript. PRP is the prion. SC is for scrapie isoform of prion protein. All kind of prions are described as PRP-SC. And you'll be, probably all of you have heard about this and studied probably in pathology. This PRP-SC is an isoform of a normal cellular protein. And the normal cellular protein that every one of us we are having, all animals and the humans, they have this particular protein which are more present, concentrated in the brain tissues, particularly the nerve tissues, and it is designated as PRP-C, cellular prion protein. This one, PRP-C. PRP superscript C designate it is a cellular prion protein. It is absolutely a normal prion protein. Every one of us, we are having this particular protein in our nerve tissues, abundantly present in our brains, inside our brain, all the astrocytes and all these and nerve cells that are present in our brain, which is essentially needed for a healthy environment, the healthy functioning of the nerve tissues. There are certain experiments in mice where experimentally this prion protein gene was prion protein gene, which is located in a chromosome 20 in human and chromosome number 2 in mice. In mice experiment, experimentally they have knocked down that particular gene, prion protein gene, and they found that the animal is showing some behavioral changes, particularly associated with dementia and normal locomotor motor functions, and gradually the animal, they suffer and they die. So this particular gene, it's a part of it's a siloglycoprotein family. Siloglycoproteins are abundantly present in all body cells. As you know, certain viruses, they target the siloglycoprotein as a receptor for entry inside the host cells. But these prions are also a category of siloglycoproteins, but they are more prevalent in the nerve tissues, particularly in the brain. And basically what happened on abnormal protein, the scrappy isoform, you understand the isoform, this is the structural conformatory change. This is a defective protein. And this defective protein, when they entered from outside, now question arises how they can enter in our body. So it has been found that either through contaminated injections or through food. That's the most worried point. This particular protein through food can enter and can get absorbed from our intestines. And these abnormal proteins, when they will reach to the brain tissue and come in contact with a cellular partner, PRPC, then this PRPC start converted to PRPSC. This is an abnormal protein and a lot of conversion takes place. This is some sort of a conversion. It's not that the PRPSC amplify inside the host and just like the viral infection, it happened from one virus, million of viruses are produced. It's not like that. PRPSC can convert the normal cellular prion protein PRPC into an abnormal one. This is some sort of a conversion. Presence of this can convert until we need to understand how this conversion occurs. And that too also, these are some sort of specificities there, like the scrappy agent. The scrappy is present in the seed for many years, and people are eating the seed meat and the brain tissue, everything we are eating, but human are not suffering. That means this PRPSC is again having some specificity. It has been seen in the same species. If transmissions occur, then the disease development takes place. However, there are certain exceptional things, but we could see it in the mad cow disease that hit the European country starting from UK in 1986 and very badly affected the people. And you will be surprised to know that the disease has a very long incubation period, which may goes up to 30 years. So that's other worry points. And globally, the disease caused by the prions are always kept in a high priority, and a lot of trade restrictions and a lot of precautions people is to take it to prevent the spread of prions to human. Okay, so try to get the basic concept that the prions are normal cellular proteins. In case of human, it is the prion gene is coded in the chromosome number 20, and they produces a siloglycoprotein group of molecule, and this is essentially needed for a healthy neuronal function in an individual. So all of us, we are carrying this PRP, and that is called as the cellular prion protein, PRPC. But when from external source is the abnormal form, the isoform of prion protein, the isoform of PRPC, which is called as PRPSC. SC designated as scrappy agent because the scrappy is the ancient disease identified in the sheep, and now it has been established it is because of this abnormal protein accumulation. So this PRPSC, once they come in contact with PRPC, then that leads to conversion of all the normal PRPC protein in our nerve tissue into an abnormal one. And when the more and more abnormal protein go on accumulating in the nerve tissue, there is a gradual degeneration of the nerve takes place. Gradually, the nerve cells will die. But very interestingly, it is not an inflammatory reaction. It's not a type of tissue injury. Gradually, they die, and even not an apoptosis process. They simply degenerate, and that leads to formation of certain pores in the brains. This is in sections of the brains where we can see in the histopathological changes of certain pores are formed. That leads to a sponginess of the brain tissue, and it is called as the spongiform encephalopathy. This is the general terminology that we use it to in all kind of prion diseases, spongiform encephalopathy. And as a result, the animal, they die. It is not a recoverable disease in animals as well as in the human. Okay, so this is a very, very characteristic things. And when we come to the what is in prions, what are the characteristics of the infectious prion? It's not the normal prion, the PRPSC. I'm talking about the PRPSC. If you go through these points, then you will understand. Please note it down this point. This PRPSC is highly resistant to heat inactivation. This is a frequently asked question. Please note it down these 7 points. They are highly resistant to heat inactivation. That means the normal PRPC protein, which is immediately they will inactivate it when you hit it, but the scrappy as in PRPSC, the prion, infectious prion protein, they are highly heat resistant. Even says that autoclaving also they can escape. I mean, there won't be any changes in the proteins. They will remain intact. Then their resistance to high concentration of formaldehyde, glutyraldehyde, all this kind. Generally, all the proteins, they get denatured and inactivated in presence of this kind of chemicals, but prions still remain active. Resistant to antiviral, obviously, this doesn't have any virus-like characteristics, antiseptic, antibiotic, etc., etc. Even they are resistant to UV, ionizing radiation, ultrasonication. So we can say they are extremely stable proteins in the environment. And this particular point, I think I have missed this one. You can write it down. It is resistant to all nucleases as well as proteases. Please add the word protease here. This is an important point in diagnosis of prion diseases. Nucleases, obviously, the prions are proteins only. No nucleic acids, you'll get it in the prion. And proteases. Protease enzymes generally they degrade the protein, but because of its very stable isoform of the normal prion proteins, they are resistant to most of the protease enzyme. So this is something like impossible to destroy this particular protein. It's remain in the nature. Okay. So prion induces no immune reaction. This is a very interesting phenomenon. If you unfold this prion, normal cellular prion, then see the amino acids of the normal cellular prions and then the abnormal prions, then you'll find the amino acid sequences are similar. Of course, recently certain studies of some variations has been seen, but not up to that extent that this amino acids will differ. Okay. So their amino acid sequence of the normal siloglycoproteins that we call as the PRPC and the amino acid sequence of abnormal isoform PRPSC is same. Only the difference is the conformation. The structural arrangement is different. The secondary structures of that proteins are different. We'll come to that point. So the prion induces no immune reactions. Our immune system cannot recognize it as a foreign. As a result, no immune reactions are generated. So there's a normal serological test that doesn't play any role in diagnosis of prions. So we'll come to that point in the later slide. Prion disease requires a very long incubation period. As I say that certain very specific study on a particular disease called as Kuru disease will come in the human. It has been found that after 30 years also the cases are coming up in those affected areas. So it's a very long incubation period. Okay. So if you understand these basic things, it will be enough for you at this point. Like this is the actual things that what it happened. Okay, so there is conversion of the proteins from the normal proteins to the abnormal one. This is that picture is showing. This is showing the abnormal proteins, and this is the normal proteins. So when this abnormal protein PRPSC through food or through certain blood transfusions or through certain injections, that is also important. We'll come to that point again. This particular protein enter in our body and get it absorbed, and when it reaches to the brain tissues where they will encounter the PRPC, the normal protein, then they will start converting. This normal protein start converting into abnormal. As a result, a lot of abnormal protein go on accumulating in the brain tissue, and that's a very gradual process. So it takes long time to develop abnormality in those brain tissues. Okay, this pictures we can explain it how the prion proteins can cause disease or the pathology. Okay, now if you see this conversions means the normal to the abnormal, then you'll find the secondary structures of the abnormal protein is more the beta conformation than that of the alpha conformation. It is only the conformatory changes, like yeah, it's here the secondary structure is dominated by the alpha helixes. Okay, now here we can see, yeah, this is one. This is the normal prion proteins, where this is the abnormal one, where the beta helix is more prominent than that of the alpha helix. This is the arrangement of the protein. We say that this is the conformational changes that takes place. It is the beta conformation, and because of which the normal protein get converted to the abnormal. Hopefully all of you are attending and listening uninterruptedly. Is there any interruptions or breaking of voice or something in the slides? Hello? Hello?
Yes, sir.
Could you see the slide properly?
Yes, sir.
Yeah, if you have any queries, any questions, you can call me in between, okay? So these are some of the diseases, the short list only, I'm just placing it here. Yes? Hello? Yes, do you have any?
Yes, sir.
Yes, what? Any issues? Can I continue? Hello?
Yes, sir.
Yes, sir.
Okay, thank you. So the scrappy, as I said, the scrappy is described in the sea populations. Even 17th century itself, there is report of scrappy description. But most of other diseases, even in the human, it was in the later part of 19th century only. Something like the Kuru disease, as you know, this is the first identified the prion disease in human that occurred in the Kuru tribes in Papua New Guinea. And the bovine spongiform encephalopathy, BSC, which is also shortly we call it as a mad cow disease, that hit in 1985 and 86 in UK, and thereafter this has become a major concern even today also globally. This is a major issues, how through beef, particularly the brain tissues, this the scrappy agent is transmitting to the human. So in mink, also transmissible mink encephalopathy, this particular condition has been detected, and these are some of the diseases recorded in humans like CDZ, ZSS. Okay, a little bit information about this kuru infections in the Papua New Guinea. It was a story of 1957 and 268. Two British scientists, they observed, they went actually for some anthropological data collections in Papua New Guinea, it's a small island near New Zealand. They want to study about the Kuru tribes, and they found that the peoples, they did not live for a long period. So most of them, they die at an early age. And then women and children, and as well as the elderly peoples, they die more earlier. I mean, rapidly. They are the main susceptible people seen in the Papua New Guinea, and they found that there is an procedure. I mean, like practice of ritualistic cannibalism. Anyone they die in the community, the brain tissues are taken by the people, and later on they educated the people, and when they abandoned this practice, dramatically the number of this death has reduced to substantial extent. But still, even now also after 30 years of that particular episodes, certain cases were detected in the tribe. Now these tribes become civilized, and they have abandoned those practices. And it was at that time they could not explain why it is so. And now after the Kusiner's concept of infectious protein particles, and now it became clear that it is an prion disease, and it has been called as the Kuru disease. Similarly, some Creutzfeldt-Jakob disease it has been detected in the European country initially, and later on throughout the globes. The disease has been detected. So these are some of the neuroencephalopathic conditions recorded in the human, and now we can correlate with this prion infection. So as you said that in 1986, the first confirmed case is called cow 133 was present to the health authority, and that animals show the signs of all the neurological signs and symptoms, and the animal they died. And when it was explored, they found that this is certain prion proteins are accumulating into the brain. And when the investigation is done, how it has come to those animals, then it was straightway the focus was go on to the incorporation of bone meal in dairy animal feed. And those bone meals were supplied from sheep slaughtering house, where the sheep brains and other tissues as an offal was converted into powder forms and were incorporated into the cattle feed. And as you know, in the 70s and 80s, there was an increasing research going on among the nutritionists all over the world, animal nutritionists, and they found that if you incorporate some animal food, the proteins to the dairy cattle, then the production increases. And it was extensively used in all European countries. Even the UK, they supplied a lot of this cattle food to other European continents, and gradually many cases of BSE was identified throughout the Europe. Alarm was set up in the US and other, but still it has crosses this Atlantic and reaches USA, and the disease become very common, and many animals, they suffer from the bovine spongiform encephalopathy. And the root was traced out to the incorporation of bone meal prepared from sheep offals, from the sheep slaughterhouse product was incorporated. And immediately they put a ban. Now totally it was banned, the incorporation of animal food to the animal proteins to the dairy cattle. And that was the source. But later on, this was spread to human. That become a more serious concern. How this particular agents can cause the same kind of disease in human, and still that outbreak is going on. We'll come to that point again. So coming to the scrappy agents, as I said a long time back, this was described, but nobody knows about the causative organism. So in 1935, another remarkable development took place where in Scotland, around 1500 sheep, they developed the scrappy-like disease. And when they trace out the history, they found that all of them has been inoculated with the formalin inactivated vaccine prepared from sheep brain to prevent the looping ill disease in sheep. Looping ill was not in our syllabus, that's why we are not discussing. It is a flavivirus infection, Flaviviridae family. It's a flavivirus genus, looping ill disease in sheep. We have not discussed about this looping ill. So to control this looping ill infections, in 1935, they have vaccinated sheep with formalin inactivated looping ill vaccines, which was prepared in the sheep brain. By injecting the sheep brain, the brain tissue was collected and it was inactivated by formalin and was injected to the sheep. And 1500 animals, sheep, they developed this scrappy-like disease. And then it was later on exploded because of the vaccination. Of course, people could not get idea why it is so. The causative virus or other things could not be explored at that time. Now, after the identification of prions, it can be correlated and disestablished. So every time when this kind of disease occur, they simply go for histopathology of the brain and they can see the spongiform encephalopathy. So this is again prevalent in the sheep and mainly through oral route. The sheep, they're picking up these infections, and particularly from infected animal in grazing area, they can pick up this infection. And this is a non-inflammatory, non-demyelinating conditions that's seen in the sheep. And because of this nervine involvement, the animal, they exhibit tremors, incoordination, intense pruritus, itching, and finally the sheep, they die. Okay. So now it can be diagnosed by the histopathology and the Western blotting technique. We'll discuss later part about it. Then coming to this mad cow disease, the bovine spongiform encephalopathy, it is a great concern and having the zoonotic importance. So after this detection in UK, it was later on detected from almost all countries, and it was directly correlated with incorporation of the sheep offals into the cattle food is the main reasons. Once it was stopped, and globally it is nowadays there is a ban of incorporation of animal protein in the dairy cattle feed, and substantially the number has reduced, and the disease never seen in the outbreak form as of today's date. But however, as you know, is having a very long incubation period. Time to time the animal, they exhibit this disease, and by coming to the food chains, the disease has also been seen in human. Okay, so the cow, they develop the restlessness, incoordinations. These are some of the timelines how gradually the disease reported from the European countries. And this is a map, of course, this is 2006, 89-2006 world map where the geographic distribution of BSC confirmed cases, which has been shown it here. This again, the OIE map of 90s only. So if you try to understand how the transmissions occur, then you will find that this is mainly spread by ingestion of BSC contaminated feed. No direct contact or lateral transmissions has been identified, or even through in utero transmission also has not been confirmed. And it is mainly because incorporations through food chain, it goes to the human as well as in animal. Similarly, there is another studies which again correlate with this surgical instruments as well as the vaccine. This has become a point of concern for all of us. Suppose if you incorporate, most of the viral vaccines which are grown in the cell culture and are used as live attenuated strain vaccines or even the kill. So we use a lot of bovine serum as an growth factor in the cell culture, and bovine protein is used in the preparation of vaccine. And that if that particular bovine serum, fetal cough serum itself is contain the prion agent, then through the vaccines, through this growth hormone injections, through these insulins and other protein extracts from the animal proteins, may be a source of infection in human. As a result, globally there is a great concern to reduce the incorporation of animal proteins into this. As a result, what happened even in our country also these days, like in bacteriological media, we use certain animal proteins, and there is an increasing change of shifting from animal protein to the plant proteins. Nowadays, most of these bacteriological media prepared for vaccine production, bacterial vaccine productions, are based on plant proteins, not the animal proteins. Okay, so some sort of a high-res agar media are available nowadays, and people are more utilizing those kind of things. And vaccines are preparing in serum-free media, where the chances of entering these things are very, very less. So this is another aspect we must consider while studying these particular infections. Okay. Some of the clinical signs and symptoms. And terminally fatal disease, the animal they die once these neurodegenerative changes that occur into the animal. Now we'll talk about this diagnosis purpose. Of course, clinical signs and symptoms doesn't give much information about this. Many diseases we could see this kind of symptoms, but certain things like non-inflammatory, no fever is produced, and blood picture is almost normal, doesn't give any indication about the infectious origin. But in the dead animals, if you go for in the histopathological sections, then this spongiform encephalopathy can be detected. Now question arises, can we go for PCR? Absolutely not, because the prions, they doesn't have any nucleic acid. So presence of the prion cannot be detected by any molecular method. So it is recommended, nowadays it become mandatory globally, particularly in the beef animal, they have to collect a sample from the base of the brain, this particular highlighted area, and that particular area is used for histopathology and as well as detection of the abnormal protein by Western blotting technique. But again, this Western blotting technique, certain international company, they are preparing some test for Western blotting test. As you know, there is no antibody production against this prion protein. So what it is done in the Western blotting technique is first digest the tissue with the protease enzyme, and then it is the SDS phase, the proteins are separated. And after separating the protein, using the antibody against the normal prion proteins, the Western blotting is done. And this is the picture. Finally, I will conclude it here. This is an ideal picture of a normal animal. And this is the extracted protein from that particular area that previously I have shown you at the base of the brain, that area. It's a normal animal without having any prion infections or disease. So this is the proteins without any protease digestion. We could see the separation of proteins and different 40 to 30 kilodalton sizes. And when in this particular land, these tissues are digested with protease. So all the proteins are digested. That indicate this is a normal prion proteins. Okay. Similarly, this experiment was conducted in tonsil material also. In the tonsils, this is the undigested prion proteins, which could be seen. But after protease digestion, totally it get digested. So after this is the picture of Western blotting. This normal proteins are showing it in the Western blotting developed slide. And here it is the Creutzfeldt-Jakob disease, the variant Creutzfeldt-Jakob disease in human, where the brain tissue, this is the prion protein, undigested, and this is the digested prion protein. You can see there is very little digestions and separation. We could see all the proteins are not digested. So we could detect after protease digestion also this particular proteins. And here it is in the tonsil tissues. After digestion, the protein can be detected. Okay, whereas in the normal animals, in the normal prions, you cannot see this after protease digestion. So protease digestion and subsequent Western blotting is the recommended test for diagnosis of prion infection, and now it become mandatory in the European continent as well as in the US. So any animal that goes to the food chain must be tested by this particular technique. Only then it will go to the food chain. So that's about this prion disease. In India, of course, there is no record about this disease, but that doesn't mean that disease will not be here. Probably it could not be detected in animals or in human. Don't know the reasons, but in the literature search, we could not get the disease prevalence in India. But most of the country, they have detected. Probably this is one reason. It's not an easy technique to detect the prion agents in the infectious tissues. However, histopathologically, we could see this spongiform encephalopathy condition. As such, the outbreaks, the clinical manifestation of mad cow, where the animal saw hyperexcitations and all the signs of nerve degenerations could not be seen in outbreak form in our country. So this is about the prions, the general properties of the prions and prion diseases. Let us recollect what we discussed it. So we talk about this particular agent. So it's difficult to say it's organisms. So even simpler than that of the viruses. So viruses, at least they are having genetic material, maybe DNA and RNA, any one, but still they are having some genetic material, and they can causes transcriptions and genomic replication, new virus particles they can form. So to some extent having the similarity with living organisms to some extent. So, but when it comes to the prions, it is difficult to term and correlate with that category. Can it be a separate domain in the life or what? Still, it is not very clear. So these are certain proteinaceous particle. So this proteins, abnormal proteins, the abnormality occurs in the conformation of the protein. The amino acid sequence, everything remains same, but only the conformation changes from alpha dominant normal proteins to an beta dominant seed are produced in these abnormal proteins, and they become highly resistant to most of the disinfectant, heat, inactivations, then etc., which can destroy it, but they can resist. They cannot elicit any immune response. This is another very important feature of prion proteins. And they are very stable, and mainly they are transmitted through food chain. So food contaminations, the scrappy agents may enter, and when they reach through as a nutrient to the brain, then conversions of the normal PRP protein takes place, and that leads to accumulation of the abnormal proteins, and gradually, in a very gradual process, the nerve cells deteriorates and neurodegenerative changes that occur. occur in the animal as well as in the human, and that leads to terminal fatality. Finally, the animal, they die. So in the diagnosis process, we don't have any other serodiagnosis method. We don't have any molecular method for detection. It's only based on protease digestions of the clinical sample collected from the base of the brain where the spinal cord meets, and subsequent SDS phase is performed, and Western blotting test, we could detect the normal one only. Abnormal proteins are not detected. So this is about this prion infection. Now this is the last slides about the viroids. As I said, that the viroids are not associated with any animals or human infections. Probably all of you will recognize this kind of potatoes we see in the market sometimes. And this is a picture showing the potatoes infected with potato tuber spindle disease. This is a viroids infections in potatoes. So 1971, Theodore Diana, he identified this particular agent, and these are certain RNA, closed circular RNA, doesn't have any capsid proteins, simple RNA. They are called as the viroids, virus-like particles or viroids, not virus-like particle, virus-like as in the term is a viroid. So in plant, particularly in tomatoes and many other plant species, this viroid infections were identified, and they can cause some disease conditions in those plants. So their pathology and other we are not interested, but for informations, I'm just putting these particular slides, and these are the example of viroids. Okay. So I guess this will be the end slide. Yeah.